We are approaching our scheduled time, so let's go ahead and get started. My name is Jessica Vionis. I'm the marketing coordinator here at Golden Helix, and it is my distinct pleasure to introduce to you Dr. Bryce Christensen. Bryce fills two roles here at Golden Helix. He's both the Director of Services as well as a statistical geneticist. Bryce joined Golden Helix in 2009 from the University of Utah, where he earned his PhD in genetic epidemiology and biomedical informatics. Before undertaking his graduate studies, Bryce worked for 2 years as a data analyst at Mayo Clinic in the Division of Biostatistics. And with that, I will pass it over to him.
Thank you for the introduction. So I'd like to welcome everyone to our presentation today. Again, my name is Bryce Christensen, and we have a very interesting topic to talk about today. We're going to discuss rare variant analysis workflows, specifically when you're working in large data cohorts. Now, I realized as we were putting this together that there really is more to it than just rare variants. So I'll point out that we also will be talking a little bit about how common variants work into the equation in this type of data analysis. If you have any questions along the way, feel free to enter them in the question box in the GoToWebinar software. We'll try to answer as many of them as we can at the end of the presentation. We may answer a few along the way as well, if we have time. And I'd like to start out by just telling you a little bit about our company. We've been in business now for a little over 15 years. We recently celebrated our 15th anniversary. We are a multidisciplinary bunch around the office here at Golden Helix, but we all have the same goal, which is to create software and provide analytic services that help you get your work done. We have two major software products that we'll be talking about as we go along today. The first one is GenomeBrowse, which is our completely free visualization tool that we launched about 2 years ago now. GenomeBrowse is a fantastic tool for viewing everything from sequence alignments to VCF files, bed files, and public annotations, and really helps you to explore your data. And again, it's completely free. You can download it at genomebrowse.com. Now, the second product, and really our flagship product that we'll be talking about quite a bit today, is SNP and Variation Suite, or SVS. SVS is really a platform for management and analysis of many kinds of genomic data. Today we'll primarily be talking about the sequence analysis functionalities in SVS, but there, of course, are a broad range of other applications in genotype analysis, copy number, RNA-seq, and you can learn more about those at our website at goldenhelix.com. Now, one thing that we're very excited about and something that we've talked about a lot over the last month or so is the merging of these two great products. So we've been in version 7 of SVS since about 2009, and it's been getting better and better and better as years go by. And we are now in the process of bringing together all of the advanced visualization capabilities that we designed for GenomeBrowse, merging them into SVS, and creating SVS version 8. All of the demonstrations I will be doing today will be done in version 8 of SVS. Version 8 is available for any of our current customers who want to try it out. It's in kind of an early release mode right now. And we're, again, really excited about it. And hopefully you will see some of the utility and some of the improvements over version 7 that we've made as we go along today. Now, many of you probably know that we do a lot of these webcasts. And the most recent one before today was given by Autumn Lofbaum about 2 or 3 weeks ago, where she talked about sequence analysis on a smaller scale when you're working with just a few samples, and in particularly she discussed family trio analysis. That webcast is available online now at our website at goldenhelix.com, and I would encourage you to go and take a look at that if you haven't already listened to it. She gives a lot of good background on the sequence annotation and filtering tools that are available in SVS. We're not going to have as much time to discuss those today as we might have. So again, take a good hard look at that. Now, as we go along today, I'm going to really try to talk about the issue of rare variant analysis and really sequence analysis in large cohorts from the perspective of somebody who has experience in GWAS, somebody who has maybe been involved in GWAS analysis for many years, has a large cohort of subjects that they want to learn more about, and is considering jumping into sequence analysis now with their GWAS samples. And so our agenda is going to go something like this. We'll start out by defining the problem. What is rare variant analysis? We'll briefly review some of the upstream data processing considerations that you'll need to think about as you're getting into sequence analysis. Then we're going to move on and talk about some of the various rare variant analysis approaches that are available right now, talk a little bit about how they've developed over the years. We'll then discuss how you can design workflows in SVS for next generation sequencing data, and then dive right into an interactive software demo. We'll look a little bit both at GenomeBrowse and at SVS 8. In SVS, we'll look both at some data exploration options as well as some of the specific analysis workflows that you might want to follow. And then if we have time, we might circle back and talk a little bit about exome chips, because that is a kind of a hot question that people have been asking us about lately. So here's the problem. Array-based GWAS for really a decade or so now has been the primary technology that people are using in gene finding activities when looking for genes that are associated with a particular disease or other trait susceptibility. Of course, GWAS arrays are really biased toward identifying common variants, and they've been very productive in that regard in identifying common variants that are associated with various traits. But as we look now at NGS technology, and especially whole-exome sequencing as one of the primary drivers, it's possible to include rare variants in the analysis. And of course, we know that individual rare variants lack statistical power for standard GWAS approaches. That's one of the main reasons that typical GWAS workflows filter out variants below a certain frequency, is because there's simply no power there to identify trait associations. But we've got all of this wonderful rare variant information coming from our sequencers, so how do we use that? And the proposed solution, and people have been experimenting with this really for several years now, is combining all of those rare variants into some kind of a logical group, whether that be all of the variants in a gene or some other functional unit, and then analyzing them together. And this is often referred to collectively as collapsing tests or burden tests. Now, those of you who have heard me speak before know that I occasionally like to go back and revisit the vault and see where we've been and where we're going. And in early 2011, so nearly 3 years ago now, I gave a presentation that included a few thoughts on this same topic. And at the time, something that was really fresh on my mind was the GA17 workshop. So if you're familiar with the genetic analysis workshop, you know that it's an event held every other year, and researchers come together and everybody does an analysis on a common data set that is usually simulated to represent some of the more popular questions being asked in the field at the time. And at GA17, it was one of the first times that the workshop used a data set that was based on rare variant analysis in a large group of samples. And the lessons that I observed from attending, and that was in 2010, in the fall of 2010, that when you have a hammer, everything looks like a nail. And what we meant by that was that at the time, people who had a lot of experience in GWAS were looking at this rare variant data and trying to find ways to shoehorn the data to fit the models that they were used to with GWAS. And it didn't always work out very well. And rare variant collapsing methods at the time were still, I would say, in more of a developmental state. They were gaining popularity, but not everybody really knew what they were about and how to use them. And my observation then is that they were a blunt instrument, but effective. They worked well if you had a really strong, obvious effect, or with a lot of samples, but maybe lacked some of the precision that you might like to see. And one of the biggest lessons that I came away from is that sequence annotations are extremely important. And what I observed at the time is that there were people who were coming in from a bioinformatics background who knew how to use things like SIFT and polyphen, where a lot of other researchers were coming straight out of a GWAS environment where really they were not quite so familiar with the different prediction algorithms and public data sources that might be used for data filtering. And it was really a learning experience for everyone. And I think all of those lessons now have been very well taken to heart by the community, and we've really caught up and moved on. And the question is, what have we learned since then, and where are we at now? And I'd like to start at the very beginning here with sequence analysis. This is a slide that many of you have probably seen before, or at least something similar to it, where we talk about the different stages in sequence analysis. And we like to break it down into 3 categories: primary, secondary, and tertiary analysis. Primary analysis largely is done on the sequencing machine, and typically results in something like a FASTQ file where we have all of these short sequence reads together with base-by-base quality scores. Secondary analysis then is the process of taking all of those short reads, aligning them to the genome, identifying where there are some variations, so where there's a SNP or an insertion, deletion, or any other type of polymorphism, and perhaps doing some QA and QC on that before moving on to the tertiary analysis, which is really trying to make sense of it. It's the whole process of visualizing variants in genomic context, annotating, filtering, calculating p-values, looking for actual trait associations. And with our software tools at Golden Helix, we really focus on the tertiary analysis phase. But it's still important to take just a minute and talk about secondary analysis considerations and what you need to think about. And Autumn talked a little bit about this in regards to small n workflows or smaller sample sizes recently. But with large n, there's really one thing that I really want to emphasize in regards to secondary analysis, and that is to be consistent. We talk to a lot of people here at Golden Helix who come to us looking for analysis tools. I have the opportunity to talk to a lot of them, find out about their study designs and what their needs are. And there's a particular conversation I've had several times. I wish I had $1 for every time I've heard this. But people will say, well, we thought we would just sequence all of our cases and use borrowed controls in the analysis. And, well, now we're sequencing controls. It's something that I've seen over and over. If you don't have your data processed following similar pipelines, you are pretty much destined to have technical artifacts that show up in the analysis process. This particular slide is emphasizing the fact that with different variant callers in NGS sequencing, you can get a lot of different variant calls. Even if you take the same alignment data, push it through 5 different variant callers, and look at the entire universe of what gets called, maybe there's a 60% overlap where they're all identical. But it's not just the variant callers. If you move upstream and look at everything starting from exome capture, moving on to alignment and indel realignment and all of the different phases before you even get to variant calling, we've seen a lot of situations where just swapping out one of those upstream steps will result in technical artifacts that are expressed at the variant caller stage. And if you're not watching out for it, it can really influence your analysis. So I can't say it firmly enough. You need to make sure that everything is done very consistently when you start looking at rare variants in sample groups. Now, with this slide, I'm not going to go into too many details. I borrowed this slide from Gabe Rudy, who gave a great talk on all of the different considerations in alignment and variant calling and so on in secondary analysis. And he got a lot of this information from Nick Lohman's blog and from another paper. But what I want to emphasize here is that when you look at, excuse me, when you look at confounding factors, a lot of them are platform specific. So I've highlighted those in yellow. And you get over to the different kinds of things that can happen in the analysis stage. A lot of these are also specific to the actual aligner and variant callers you use. So one more time, I'm just going to emphasize, you need to make sure you're being consistent to make sure that you don't have technical artifacts showing up in your data where there's certain types of rare variants that are just occurring in your cases or in your controls by chance. So the bottom line, no matter how much you want to use 1,000 genomes as your control sample, or no matter how much they want you to use their data for controls, stop and think about it a little bit before you dive in too far. And if you do decide to use public control data, just keep your eyes open and watch for anything like this to happen. Okay, so that is my warning on secondary analysis. And let's go ahead and jump ahead and talk about tertiary and some of the sense-making issues with large sample sizes and rare variants. So there's really two primary approaches that we see people taking with sequence analysis. The first one being a direct search for susceptibility variants. So if you assume that there is some highly penetrant variant, perhaps with evidence of Mendelian inheritance, and you know it's in there, you might just dive in and start digging around looking for rare novel variants that are predicted to have extreme functional consequences. Maybe you know what gene set or what pathway to look in. It can be extremely successful in some cases. Sample sizes there are usually small, single cases, nuclear families. It's the kind of workflow that Autumn was addressing with the last webcast. Now, there are also situations I've seen where with larger groups of samples, you might also want to do the same thing. Maybe you have several children that have a similar disease, but you weren't able to sequence their parents, so you don't have the trio structure. You might want to collect them all together and see if you can find something in common between them. And I'll show you some ideas as we go along for how you might do that with large sample sizes. But really, the most common approach, and we've already talked about it a little bit, is this idea of rare variant collapsing. And it's, of course, more common in complex diseases and may require very large sample sizes to get statistical power. And I'll just make sure we're being really clear about that. I talked to somebody, I can't remember if it was earlier this year or late last year, but someone who's involved with a project where they have about 6,000 samples. They're looking at complex traits using a lot of different rare variant collapsing tests, and they've determined that their study was underpowered. So we talk about with GWAS and common variants that to really unravel some of these diseases, we need samples in the tens of thousands. We're finding that with rare variant collapsing methods, we need similarly large sample sizes in many cases. But the whole concept of rare variant collapsing is that if you assume that any of several loss of function variants in a susceptibility gene may lead to the same disease or trait, then you can devise an algorithm that will look for that pattern. There's a lot of different algorithms, different statistical tests that have been put forward for doing this. Even though it is much more of a statistical approach than that direct search in the first bullet, it still tends to rely a lot on bioinformatics. So there's two major families of collapsing tests. And when you look at how this slide is organized, it's approximately in chronological order, with some of the earliest methods at the top. And burden tests, which are perhaps the simpler of the families, tend to combine minor alleles across multiple variant sites, usually within a gene. But some of the earlier tests did this without doing any weighting. It was really just a straight up, let's find out how many minor alleles are in cases, how many are in controls. And some of the methods for doing that, CMC, CAST, CMAT, you might have heard of some of these. From there, we moved ahead a little bit and looked at models that weight variants based on allele frequency, and then got a little smarter and learned how to use weighting schemes that adapted to the data, or thresholds that adapted to the data. Thresholds meaning, what do we define as rare, what do we define as common? But one of the major questions that has come up over and over again is, well, what if, and not even what if, it's pretty much fact that when you have a large number of rare variants in a given gene, many of them are probably going to be completely benign. Some of them may even have protective effects. So how do we account for the fact that effects could go in either direction? And then there were some methods developed to address that. More recently, the kernel type tests, particularly SCAT. SCAT is kind of the flag bearer in this area, that allow for individual variants to have effect in either direction. They also permit covariate adjustment based on kernel regression. If I understand the history on these, a lot of it started with this 2008 paper that described doing region-based tests with kernel regression based on tag SNPs. That was adopted into more of a rare variant framework by SCAT. Since then, there have been a number of extensions to the SCAT method, SCATO being one of the most broadly adopted. Now, you'll notice that I highlighted with bold text a couple of these methods up at the top, CMC and KBAC. And I highlighted those because they were the first two methods that we adopted into SVS, and they're the two methods that I'm going to be showing you today as we go along. CMC, to tell you a little bit more about it, is a multivariate test. It's a simultaneous test for association of both common and rare variants in a gene. It has some flexibility in how you determine what's common and what's rare. But it's a very flexible and useful test in many ways. KBAC is strictly a rare variant test, but it models a kind of a multi-locus genotype within each gene, and has an adaptive weighting procedure that goes into the model, and it essentially tries to identify which multi-locus genotypes may be associated with higher or lower sample risk. And again, there are a lot of options on how you use it and the way it's implemented in SVS, and we'll take a look at a few of those as we go along. Now, within SVS, I want to emphasize that SVS is very flexible in workflow design. It's not a black box in any way. It's really a platform that has a wide range of tools that can be used for data manipulation, variant annotation, variant filtering, calculating association tests. And as long as we know what it is we're looking for, we should be able to construct a workflow using the existing tools that will get us to that final goal. So are we looking for a rare non-synonymous variant with a dominant Mendelian trait? We're looking for a gene with excess rare variation in cases versus controls, or even perhaps in comparison with a quantitative trait, so excess rare variation with higher values or lower values. So as long as we know what we're looking for, we can usually put together a workflow that will get us there. Now, one of the features in SVS that allows this incredible flexibility is Python integration. And the Python interface is something that most of our customers probably don't get involved with directly, but indirectly it's unavoidable. The Python integration in SVS allows rapid development and iteration of new functions. A lot of the existing functions that our users use on a day-to-day basis were originally developed using the Python API, which allows us access to not only SVS functions, but also to extensive analytic libraries. And so we can use this to build a function, send it to a customer, they can drop it in a folder and run it immediately. We can iterate back and forth. And if you happen to be a Python ninja yourself and want to learn how to do some of this scripting, it's all documented in the manual. But one of the great things about it is that it allows us to keep this repository on our website of useful scripts that have either been written for a customer with a particular need, or it's been written when we recognize internally that we need something in order to finish a job that we're doing in one of our services engagements. And if you go to our website, goldenhelix.com, you can find this repository and look through it and get an idea for some of the useful little tools that are in there. They're all plug-and-play functions. So download a little script and drop it in the specified file or folder on your computer, and it's there ready to use inside of SVS. Some of these we've written ourselves, some have been contributed by customers, but I'll emphasize that the popular ones often get adopted into the shipped version of SVS. So if you see something in the repository that you really like, let us know. It will probably turn out that a lot of other people would like to use it as well. All of the scripts are forward compatible to SVS 8, which is a very good thing. So there are two scripts from the repository that I want to feature as we go along today. One is this script called Activate Variance by Genotype Count Threshold. It's very useful with large data sets. And also this one, Filter by Marker Map Field, which is really useful to help you interact with some of the annotation variables that might be present in a VCF file. Of course, there's a lot of other useful scripts on there. I've listed some that you might want to take a look at along the way if you happen to already be an SVS customer. And if not, of course, you're welcome to get a free trial of the software and see how any of those might be useful for you as well. So for our interactive demonstration today, we're going to start out by looking at GenomeBrowse and see what you can do with multi-sample VCF files in the free genome viewer. Then we'll move on to SVS 8, and there's two workflows I want to go through. One is very exploratory in nature, and I want to do that, number one, so that we can feature a few of these useful scripts. But also, it helps to reinforce exactly what is happening in the background on some of these collapsing tests and burden tests. And once we've gone through that, we'll move over to a different project and walk through a project that's had a KBAC analysis done and a CMC analysis. Take a look at those, find out more about how they work, what the workflows are like, and take a look at the results. So with that, we'll go ahead and shut down PowerPoint for a minute and open up GenomeBrowse. So many of you by now, I'm sure, are familiar with GenomeBrowse. If you're not, we'll just take a few minutes here to get acquainted with it. So GenomeBrowse is designed primarily for viewing BAM files containing sequence alignment data, as well as VCF files and BED files being kind of the most common data formats that we run into in the open source genomics environment. We also have the option for you to add numerous annotation sources that we have cataloged. If you happen to be an SVS customer, you can also create and view your own annotation sources here. But what we're seeing at the moment is just a couple of basic annotations with the reference sequence and a gene annotation file. Here on my desktop, I have, I don't know if you can see it for sure, but I have a VCF file from the 1000 Genomes project. This is a VCF file that has over 1,000 samples in it. I'm just going to drag it into GenomeBrowse. We're going to take a look at it. Now, if you have worked with VCF files very much, you know that it is a massive text file. And Sometimes you need to be able to get into that text file and look up some little bit of information, and it's always a pain to do it. Some of these files are so big that you can't even open them with standard text editors, and once you do, searching in them can be a bit of a challenge. And so what I want to emphasize with VCF files in GenomeBrowse is that GenomeBrowse is like a free tool for viewing, navigating, and interacting with these VCFs. What I'm seeing on my screen right now, as I zoom in a little bit closer, you can see it better, is a visual representation of all of the variants that are captured across 1,092 samples on chromosome 22. And as I zoom in closer and closer, I can get to the point of seeing individual genotype calls. By clicking on any one of them, it will show me all of the underlying data for that particular genotype call. So if I'm just doing some post-analysis QA and need to go back and look up some of the quality scores on a particular variant call so that I can put it in my manuscript, you might find that this is a lot easier than trying to open up the VCF file in Emacs and search for that bit of information. So if you need to see the likelihood scores, in this case it's showing me all of the 1,000 samples of that particular variant position because it is a multi-sample VCF, but hopefully you see some value here. And of course, navigation is simple and easy. Click and drag to pan, scroll wheel zoom, and you can type coordinates directly or gene names and jump directly to those. But of course, just visualizing data, while very useful, is not the end goal in most cases. There's a lot of additional work that you might want to do to learn more about the data that's in here. So I'd like to go over to SVS now and open a project that has that exact same chromosome 22 VCF file imported into it. So for those of you who might be seeing SVS for the first time today, I'll emphasize that it's a project-oriented software tool. It's designed to help you organize inputs and outputs from a particular analysis project in one place. And it gives annotations about your workflow, so whenever you perform a function, notes are kept, and it will have kind of a logical tree-based organization of all of your results and outputs up here as we go along. Now, to begin, let's just get acquainted with this data set. I mentioned it would be the exact same data that we were just viewing from that VCF file in GenomeBrowse. And you can see, if you look at everything here, we have 1,092 rows. I can scroll to the end and see them all. We have just under 500,000 columns of data, and I can scroll out to the end there and see all of those. Now, also, there's this bright green map button jumping off the screen at me. If I click that, it opens this collapsible area of the spreadsheet where you can see a lot of those same quality fields that had been present in the VCF file. So when I clicked on that variant and we looked at the data console, we saw some of these same things, like this average posterior probability that comes from the imputation process that 1,000 genomes did, or the mutation rate, or the R-squared from the imputation. Also, there are allele frequencies from different populations. This is all data that we didn't create. It came directly from the VCF file. Now, suppose that I have a workflow where it's really important for me to use one of those values out of the VCF file as a basis for filtering. Typically, with our standard workflows in SVS, we base most of our filters on annotation tracks, like those tracks we saw in GenomeBrowse. We might use one of those that defines frequency in some population and drop out any variance above or below a particular frequency threshold. But what if we already have the data here and we want to use it? That's one of these useful scripts that I mentioned from the script repository. And I have it here in my drop-down menu. It's called Filter by Marker Map Field. It's a very simple descriptive name. And what this one will do is allow me to select a field to base a filter on. And for the sake of demonstration, I'll use that R² field. And this is capturing the quality of the imputation that was done that was part of the 1,000 genomes workflow process. And let's suppose that I want to set just a very strict threshold and only get the very, very highest quality things here. Practically at this level, you're only going to get variants that were directly sequenced as opposed to being imputed. But I'll say 0.9 R² and inactivate anything that's below that threshold. Let's go ahead and run this. And so now it's scanning through, going column by column, looking at that R² field. And when it's done, we'll see that anything that failed is going to be inactivated. So it indicates about half the data was inactivated. And we can look here and see columns have been turned gray. That indicates they're turned off, won't be used in the next function that I apply. Of course, I have the option to create a data subset of just the active columns, which I did. I'll rename this so I know that it's R² greater than 0.9 and go on. And you'll notice as we go along, we're going to continue to build out a tree of all of the outputs and subsets that we create. Now, of course, most workflows we want to look at this data within the context of some kind of phenotype data. And I have another spreadsheet here with some simple phenotypes. Really, I just have a simulated case control variable in which major population groups they represent in the 1,000 Genomes project. Let's go ahead and merge these together. Merges just take 4 clicks in this case to bring data together and get it all in one place. And now let's suppose that I'm working on some kind of a population genetics question where I want to know about variants that are common in one group and rare in another. If I summarize this population column, I can see that there are about 379 Europeans here, slightly fewer Asians and Africans, much smaller number of admixed American representation. But let's suppose that I want to use this population variable and find variants that represent different frequency levels in each population. This is another one of those useful little add-on scripts. And it's called Activate by Genotype Count Threshold. I'll go ahead and expand this out so we can see the whole thing. And start from the bottom. So this is asking me, group by group, based on my dependent variable, what's the threshold here that I'm looking for? And I'll say that in Europeans, I want at least 50 individuals that are carriers, so they have one of these genotypes with an alternate allele. And for that same variant, I want at least 10% of Africans also to be carriers. But then for Asians, I want to turn it around, and I say less than 10% of Asians to be carriers. And also with the admixed American groups, we'll say less than 10%. And again, we're highlighting either heterozygous or homozygous carriers. There's a lot of flexibility in how you set this up. Hopefully you get the idea that it's a useful little tool for looking for different patterns. And this is going to take just a minute to finish. And while we're waiting for it, I'm going to go back to one of the questions that I saw come in at the very beginning of the webcast. Somebody asked what QC criteria should be applied to imputed rare variants instead of R². So I briefly mentioned R² here already. Imputed variants are kind of tricky, especially rare imputed variants. If you've worked with imputation very much, you know that common variants tend to impute with pretty high quality, because if you have another common variant that has high correlation there, you can get a pretty good imputation of what the minor and major alleles should be for every sample. Now with rare alleles, of course, the imputation is a little bit more difficult because unless you have an extremely large reference population with a pretty good representation of that particular rare allele, you're not going to have a very good idea of what it is correlated with, so it's harder to impute that rare allele. Personally, in the imputation work I've done, the thing that I like to use for quality there is actually to look at the genotype probabilities, where you get a triplet of values with a probability for both homozygous minor, heterozygous, as well as a probability for homozygous major, and use the probability of the homozygous or heterozygous minor as the basis for filtering. It's really pretty similar to the data you get when you look at dosage, allelic dosage from imputation, but maybe we'll come back to that in a minute. We see that our function finished up, and we also see that there are only 512 columns that remain active. And so I can subset this, and what I should find now is these remaining columns are all going to be variants that fit that pattern I was looking for, at least 50 of the Europeans being carriers, pretty good frequency in Africans, lower frequencies in the other groups. To visually capture that, let's just go ahead and bring this into the browser. Now, this is the first time that we have seen GenomeBrowse working inside of SVS. And you'll see that the data looks pretty similar to what we saw in GenomeBrowse. But in SVS, we have a few extra tricks we can use, like using a grouping variable based on the phenotype data in the spreadsheet. So now I can recognize with this colored axis where each of my sample groups are, and it becomes more obvious that there's higher data density in the Europeans, lower density in the Asians as expected, and higher again in Africans. As we zoom in and look at it a little bit more, we can also start to see where all of our different variants fall relative to genes, other annotations. Another thing that's kind of fun is that we have the ability to use different variables as labels for each variant, and also to hide the reference allele. Sometimes when you just look at the non-reference, the color pops out a little bit better. But I can turn on the reference alleles so I can see specifically what everybody has at each position. And also, as I mentioned, I can adjust, use different labels. So I can just directly label here, what is the African allele frequency? What is the average posterior probability? What is the RSID if there is one? So there's, again, some flexibility in how this works. But now that I've identified some of these variants that are maybe common in one group, rare in another, it's similar to the workflow that I talked about if you wanted to try and directly identify susceptibility variants. So maybe if I take these 500 and, I guess it's 508 when you take out the 4 columns of phenotype data variants and want to learn more about them, this is the point where you might go into some of the classification tools and some of the annotation tools to learn more about individual variants. So if I run the standard variant classification, and this should take just a moment with the size of data we have here, I think we'll find out that very few of these variants are actually in genes, and even fewer of them are coding. And so we can see here just 6 coding variants were identified, and if we come back and look at where they all are, what gene they're in, it turns out that they're all synonymous except there's one that was in a transcript that had some issues. And so if we had something here that looked like a potential loss of function variant, we might have something we wanted to follow up on. But since we don't see any one individual variant, what if we wanted to pursue a collapsing strategy? and come back here where we had applied those filters, and we just have these 500 or so variants. There's a function here that doesn't get used a lot in SVS, and I think that it's an incredibly valuable and incredibly powerful function called count variants per gene. And when we look at this, it will give us kind of the basis of how a collapsing test or a burden test is created. What we can do with this is summarize on a subject by gene level how many variants are present in any given gene, or just the simple presence or absence of a variant, or the total number of alternate alleles present per sample per gene. And I'll go ahead and just look for either heterozygous or homozygous variants and just calculate the presence or absence on a sample by gene basis. Go ahead and run this. This will take just a moment. And we get our results here. And again, we have samples in rows, so 1,092 rows. And then each column, it's not actually a gene. It's technically done transcript by transcript. So that's why we see two different columns here with the same gene name. But we can look around and see this sample has at least one variant in that gene. This sample has at least one variant in that gene. And this is a representation that's very similar to what is happening in the background on collapsing tests. So if I merge this now with my phenotypes, I get a representation where I can look again at, say, the population variable and compare gene by gene to see if there's any one gene where maybe not always the same variant, but some clustering of variants that is related there. And if I just use a simple tool here to summarize columns, and we'll just calculate means, turn off some of these extra statistics so we don't clutter the result. Well, I'll get the sum too. What we'll end up with now is a spreadsheet where we can see group by group. Maybe we'll start with the Asians. Well, we left in our case control variable. It's also getting summarized, but we can see here on the second row where the gene is SYN3, about 15% of Asians have at least one variant there. That represents 42 total Asians. We can see that about 63% of Africans have at least one variant there, about half of Europeans. And so this turns out to be a really useful way to summarize data when you're looking directly either for an individual variant or for a gene harboring a lot of variants. But it's also very simplistic. And if we want to see a bit more of a sophisticated way of looking at that, we need to back up and open up a different project here that's actually had a whole genome workup done as opposed to just one chromosome like we were looking at there. So in the last webcast and in several of our previous webcasts, we've talked about how you can apply data filters based on different annotation sources. And we're not going to repeat the whole process here, but I just want to emphasize a little bit about how it was done. So in this particular project, again, it is the 1,000 genomes data, but we've reduced it to all of the exon variants across all of chromosomes 1 through 22 plus X. And what we end up with is about 714,000 total columns of data, all of them, of course, representing a position where at least one sample out of 1,000 has a non-reference allele. And so starting with that 714,000 variants, we proceed to filter it based on exons that are represented in RefSeq, and we see there were 435,000 of those. We then compare it to the NHLBI exome sequencing project, which gives us a report indicating variant by variant which ones are found in the database, which ones are not, what is the exact frequency that they're observed with in the database, flags whether or not they were filtered. After applying the filter, we end up with 363,000 variants. proceed then to remove anything that's considered common in dbSNP 137. That reduces the list a little bit more. Again, it produces another report. Then run it through a variant classifier where we reduce just the variants that are either non-synonymous or, I guess you'd say, non-silent. We're just taking out anything that was classified as synonymous. And we end up with about 204,000 variants across the exome that are considered to be rare and non-silent. And that is kind of the workflow you need to do prior to running something like the KBAC test, where you're focusing on rare variants, potential loss of function variants. And so if we come into this final spreadsheet where we had just over 200,000 columns to look at, and come into the collapsing methods section and run KBAC with permutation testing, I won't take the time to run this right now, but I'll point out that it is based on a permutation test. We have this adaptive permutation testing option that really speeds up the process. It lets you specify a threshold, say 10 to the minus third here, or second, where you're saying that essentially anything that it recognizes immediately is not going to pass that threshold. It just skips it and moves on. So it really speeds things up. And when you run it, what you get as a result is something like this, where you get one p-value per gene. You can take that list, sort it, see what comes to the top. In this case, I used 1 million permutations, but you can see that there are only a few genes that just 2 genes that needed the entire set of 1 million permutations to determine their significance. And scrolling through, you see there are a lot of additional statistics that are captured relative to that test. Now, with CMC, where CMC is more of a test that also includes some common variants in a multivariate testing scenario, the workflow we did was a little bit different. We started from that same set of 435,000 that are in RefSeq exons, proceeded then to classify them and reduce it to the non-synonymous variants. You'll notice that this number, 239,000, is not a lot higher than the 204 here. So we can see there's maybe 25,000 variants that are more common that made it into this second analysis. But here we are able to take that, run it through CMC. With CMC, similarly, we get results where we have 1 p-value per gene, together with a lot of additional statistics that help summarize the results. And also, I decided on this particular workflow to take that same set of variants and just run it through a SNP-by-SNP test to see what the results look like. And let's take a look at those now. And we'll begin by looking at KBAC, and we'll look across the genome. So remember, when we looked at KBAC and sorted the p-values, there was one gene that came right to the top, and that gene was ALPK1. I can click here and see the gene name, scroll down, see that the negative log 10 p-value there was, actually did it twice, once with 10,000 permutations and it peaked at 10 to the minus fourth, once with 1 million permutations down here and it peaked at 10 to the minus sixth, so it's probably much more significant than what we're seeing. It's really maxing out the number of permutations that I can do. But if we zoom in close around that one, we see it's in this gene ALPK1. And if we center our view around that gene, we see this pattern where, based on our case control variable, which is grouping according to phenotype, false meaning control, true meaning case, all of our cases are at the bottom. It's a bit unbalanced. But we can see a lot of variation down here among the cases, very little variation up here among the controls. There are a few random variants. Hopefully, on the other side of the webinar, you can see it with your screen resolution. All of these little dots are representing points where there's some variation. Now, if we add in the results from the CMC test, of course, CMC is based on a different sort of a test with an asymptotic distribution as opposed to permutations, we can look here and see that same gene is really just jumping off the charts at 10 to the minus 150th. It is simulated data. It's maybe a little bit overdone here. But in general, if we zoom back in and look a little bit closer, and I'll just adjust the range a little to see some of the lower p-values. In general, the two tests kind of agree, but it's important to remember that they are not identical tests. One is including some rare variant or some common variant contribution together with the rare. If we move over to where our main result is on ALPK1, of course, the CMC result is off the top, but if we add in the Fisher's exact test in red that's just capturing SNP by SNP, we see that in this case, there are some of the SNPs that are individually significant. And again, I'll emphasize that this is a simulated result that's maybe a little bit overdone. When you're doing this out in the wild, you're much more likely to have a scenario where rather than one significant gene, there are several mildly significant genes, where there are a lot more questions to be answered. But hopefully you've been able to see from the demonstrations so far some of the flexibility of SVS in terms of designing your own workflow, performing different types of tests, and getting you to a particular endpoint to solve your questions and your problems. Now, back here in the slides, there's just one last thing that I want to take a look at, which is, what about exome chips? The question comes up a lot. Can I use exome chips with this kind of analysis? The answer there is yes, but you just need to keep in mind exome chips don't capture all of the rare variation, and because of that, they're not quite as powerful as using whole exome sequencing, but of course they're a lot cheaper and they're incredibly useful, and I would be happy to talk to you more about using exome chips. Just a few other pointers on exome chips that we'll skip over now due to time. But in closing, I just want to let you know that we love to answer your questions. If you want to know more about the product, if you want to get a copy to try out, give us a call, send us an email. If you just want to get yourself an autographed Golden Helix t-shirt from Gabe Rudy, I'm sure he'd be happy to work with you on that too. And with that, I think we'll go ahead and take some questions here.
You know, thank you for that really great webcast, Bryce. It was very, very interesting. If you didn't have a chance to ask your questions during the webcast, go ahead and feel free to type them into the questions pane now. We are sitting right at 11, but we'll stay a little bit longer on the line to answer as many questions as we can. If we don't get to your question or you have to leave due to the time constraints, we will do our best to get back to you individually. Before we go ahead and launch into the questions, I'd like to mention that this webcast was recorded, and we will be putting it along with the slides on our website tomorrow. I'd also like to just let you know that we do have these webcasts about once a month, so make sure to check our website for announcements of more events to come in the future. All right, so let's go ahead and do some questions. So let's start with a pretty easy one. Bryce, do you have a projected full release date for version 8?
Full release for version 8, I believe we're looking at the first of the year. Of course, you can get access to it now. Later this week or first of next week, we will be releasing version 8.01. The major advantage there being that it will now be supported on Mac, which has been missing with the version 8.0 release on SVS, which has only been for Windows and Linux so far.
And tell me again how I could get my hands on it now.
Just give us a call or email support. If you're a current user, just let us know that you would like to have access to version 8, and we can set a flag on your license that will allow you to download and install it. Okay.
Going on to the next question, does GenomeBrowse only take VCF or BAM files? And can you look for RVs if you only have a CSV file of genotype data?
So GenomeBrowse itself The free version is designed to look at VCF and BAM and bed files as the primary inputs. If what you have is just a CSV file, I assume it's just some kind of tabular representation of genotypes, we would probably need you to either get SVS and import that as a spreadsheet and then view it through SVS, or maybe we'd have to figure out another way to do it. But no, I don't think it directly will read anything from CSV.
Okay, good to know. And if I had suggestions for GenomeBrowse?
For improving GenomeBrowse?
Yeah.
If you've got suggestions, we'd love to take them. Just send them to support@goldenhelix.com and let us know what you think.
Okay.
And so, I'll be clear on that one point. There are a lot of features in GenomeBrowse that are the result of customer feedback. We're probably going to send out an update of GenomeBrowse within the next few months that is going to incorporate a lot of changes that are not so much changes as enhancements.
Okay, good to know. Next question. I am wondering how SVS supports indel variants of VCF files.
So SVS, in terms of how it handles and processes VCF files, it will read whatever is in your VCF. If you have indels there, it will read them into the spreadsheet. And once they're in a spreadsheet, you can do all different kinds of analysis work on them. I'll be honest that a lot of our annotation and filtering tools do rely on publicly available annotations, and indels in general are not annotated as well as SNVs. So there are maybe a few limitations in terms of things like functional predictions, but in general, we can do quite a lot with indels.
Okay. Can we do collapsing analyses on commercial arrays instead of sequence data? And if yes, do we have any publications that support that?
So collapsing tests on commercial arrays, I assume by that question that maybe you're getting at exome chips, and I tried to answer that at the very end as being yes, if what you have is actually a regular GWAS chip. Certainly the tests will work there, but you need to keep in mind that there are some underlying assumptions. Most of these rare variant burden tests are, of course, designed for rare variants. But if you have something like the 5M or the 2.5M from Illumina that has a pretty good contribution of rare variants on them, you should be able to use those tests. One of the caveats being that the SVS implementation of those tests does expect that the genotypes have been called on the same strand that sequence variants are called on, and that may require a little bit of data manipulation before you can do it.
Okay. What is the difference between AMRAF and ASNAF?
Okay, by that I assume you're talking about the fields that we saw in the VCF file from 1,000 Genomes. That is calculated allele frequencies for different population groups. So there's the African allele frequency, the American allele frequency, and there are a few others in there as well. But
okay.
That's what it is. It's just a calculated field that had been added to those VCF files as an annotation. The VCF format, of course, is very flexible in handling lots of different annotations.
Okay, just a couple more. Are there single variant methods optimized for rare variants in SVS, e.g. fifth logistic regression, exact logistic regression, etc.?
So there are a few different types of analysis that I did not show today. Yes, we definitely have logistic regression, a few different options there. The specific ones you're mentioning there, like first logistic regression, is not implemented. But if you want to know more about the exact methods, let us know. I'm sure that probably Greta in support would be the best person to talk to about that.
Okay. Are we planning to incorporate SCAT into SVS anytime soon?
Okay, so that is a great question. And looking through the list here, it looks like several people have asked a similar question. And here's what I am authorized to say. which is that we are definitely reviewing some options for updating our collection of collapsing tests. The tests that are there were implemented in version 7.4 and expanded in version 7.5, and we are revisiting that topic right now. And if you have a particular request on the functions you'd like to see, let us know. We definitely are pursuing a few that we'd like to get in there.
Okay, and I will ask one last question for you, Bryce. Are both of the softwares, GenomeBrowse and SVS, applicable to bovine genome analysis?
Okay, great question. And the answer there, I'll actually answer with a bit of a demonstration here, which is that if you go into SVS and just say open a browser window and take a look at this list of supported genomes, you'll see a really long list, including right there Bos Taurus, several different builds of the Bos Taurus genome for cattle. But really, SVS has the ability to work with pretty much any diploid genome. When you get into higher ploidy numbers, we do have some issues with it. But in general, even if you don't see your species in this list, let us know. All we need to know is how many chromosomes it has, how long each one is, and if you happen to have access to the reference genome, it's incredibly helpful, but we can make sure that it supports whatever it is you need to look at. I noticed there was another question earlier about polyploid species, and we have some issues working with genotypes that can't be easily represented as diploid. If you have a way to manipulate that to look like diploid or look like some kind of a log additive numeric representation, then there's more that we can do. Within GenomeBrowse, any standard file format, so if you have just a standard BAM file or standard VCF, we should be able to annotate and navigate within that and work on it. But in general, we are species agnostic, is what I'd like to say.
Okay, thank you everyone again so much for coming today. If we didn't get to your question or you have more questions, please feel free to shoot us an email at info@goldenhelix.com. And thank you again for coming. Thank you, Bryce.
You're welcome.